Hey guys, welcome back to my channel, MrGamingDoc. So, sorry for the abrupt interruption before. So we are at polysaccharides. They are divided into homosaccharides and heterosaccharides. We need to be doing this very fast because we have a lot of stuff to complete. So homosaccharides, they are starch, dextrin, inulin, glycogen, sorry, okay, glycogen and cellulose. So if we look at the longest list one, which is polysaccharides, they are divided into homosaccharide. And examples are starch, dextrin, inulin, glycogen, cellulose. Homo means the same kind of sugar molecules, the same structure, and they are connected by their respective groups, and they make up long chains which are uncountable. Starch, dextrin, inulin, glycogen, cellulose. And when it comes to heterosaccharides, so I think these are all hard facts, right? We can't do anything much more about them to convert them into conceptual information because they are just very plain facts. So we can't do anything about it, but we'll be diving too deep into the next stuff. So heterosaccharides are hyaluronic acid. I'll be giving you a hint to remember how these things can be much more buried in deep in your minds. Chondroitin, keratin sulfate, chondroitin sulfate. Yes. And heparin sulfate. So if you're very observant, you could see a little difference in these things. When it comes to homo and heterosaccharides, all the things that can be produced in our bodies, they are hetero, which are hyaluronic acids, keratin sulfate, chondroitin sulfate, heparin sulfate. But when we can't produce them, they are going to be homosaccharides, which are starch, dextrin, inulin, glycogen. Oh, I'm sorry, glycogen, we can't do that. And other than glycogen, starch, dextrin, inulin, cellulose. This is my observation, just for namesake. If you have a better kind of explanation for this, you could post in the comment section below, and others may benefit from this. So again, I'm telling you, this is not just about preparing hot and cold. We're gonna play games in between just for fun, just to chill out a bit. This is to complete as fast as possible. So we need to get going with this. Okay, next. So next point is if two monosaccharides differ from each other in their configuration around a single specific carbon atom, they are known as Epimodus. So for example, they've given glucose and galactose. We're gonna create a new sticky note. So this is about Epimodus. I think we need to make this bold. Yep. Glucose and galactose. So the reason for this explanation is two monosaccharides. These both are two monosaccharides, differ from each other in their configuration around a specific carbon atom. So somewhere in it, maybe let's just see, glucose and galactose. So yeah, so where do they differ? In their fourth carbon atom. Okay, one is present to the right and one is present to the left. One is levo and one is dextro. See, see the difference here. So that's the only thing that because they resemble in their structure in each and every carbon atom except one specific carbon atom, they are called epimers. Okay, the next thing, let's go to this thing. Okay, a method for synthesis of higher monosaccharide from a lower one is called Kiliani Synthesis. Okay, Kiliani, sorry, Kilian, what's the spelling again? Yeah, synthesis. What is this about? Let's use our, what do you say, Google at our hand. Yeah, Kiliani. Did I? Yeah, sorry, I typed high. Did I write a correct spelling here? Yeah. So the thing is, the Kiliani or Kiliani-Fischer synthesis, named for German chemist Heinrich Kiliani and Hermann Emil Fischer, is a method of synthesizing monosaccharides. The new chiral carbon is produced with both stereochemistries. So the product of a Kiliani-Fischer synthesis is a mixture of two diastereomeric sugars called Epimodus. It's interesting. Yeah. So they're saying after they synthesize monosaccharides, they are gonna be in two different. They are gonna be a stereoscopic replicas of dextro and levo. So it's gonna be Epimodus again. Okay. If you still have a doubt in it, just ask me a question in the comment. We are doing a revision, so we're not going into specifics. So inversion of sugar is the process of changing optical rotation from dextro-rotatory to levo-rotatory. The hydrolyzed mixture of sucrose containing glucose and fructose is known as invert sugars. Okay, these are very basic fundamentals. So what are invert sugars? Sucrose and fructose, sorry, glucose and fructose. And what is inversion of sucrose? It's a process of changing optical rotation from dextro to levo. But when the sucrose is hydrolyzed, it gives rise to glucose and fructose. So that's why sucrose is called invert sugar. Okay, seems plain enough to me. Starch and glycogen are carbohydrate reserves of plants and animals respectively. So starch, animal reserve of plants, glycogen, animals. Cellulose is not digested in humans. Okay, maybe it's a hardcore fact since right from our intermediate. Glucose is called energy currency among carbohydrates. ATP is energy currency of the cell. Again, it's a hardcore fundamental. Glucose shows stereo as well as optical isomerism. Okay, it is a dextrorotatory and an aldose sugar. Glucose, aldose, sorry, dextrorotatory, dextro R I'll write, and stereo and optical rotation. Okay, plain enough. A glycoside containing a sugar molecule, glucose, and a non-sugar molecule. A fructoside contains a fructose sugar molecule and a non-sugar molecule. Okay, glycoside and fructoside. Digitalis is an example of glycoside. Okay, nice example actually. Digital is used in congestive cardiac failure or congestive heart failure to increase the force of contraction. Yeah, basically it alleviates the symptoms, and it's direct cardiac stimulant. Okay, I actually have a favorite technique to memorize pharmacological classifications and drugs and all. I'll tell you guys when we come to the pharmacology section. Okay. The end products of carbohydrate digestion are glucose, fructose, galactose. Okay, carbo digestion results in glucose, fructose, galactose. We are just scraping the surface of the biochemistry. There's still a lot more to read. Glucose absorption occurs chiefly by active transport. Okay, this is a good point. Glucose absorption chiefly by active transport. It doesn't say glucose absorption occurs only by active transport. It says chiefly, which means it can also be transported in by passive mechanism. But most of the glucose is absorbed by actively. So blood sugars values. Fasting, they say, this book says 80 to 120 mg per deciliter, but actually according to most of the textbooks I think 70 to 110. Random 110 to 150 mg per deciliter. Postprandial, which means after food, 150, I mean close to 150 maybe, or let's give a take less than 150, sorry, mg by deciliter. So these are all maybe outdated values. They get updated many times, but let's just see in the internet. Okay, range is in non-diabetic fasting 70 to 110 as I said, but less than 40, less than 80. Okay, it depends. Hypoglycemia, let's go to the sticky notes. Where is our, yep, this, yeah. Hypoglycemia, what does it say, is due to decreased blood sugar levels less than 65 mg per deciliter. Hypoglycemia is increased blood sugar levels greater than 120 by deciliter. It's hyper. I don't know. Let's see on the internet again. Okay. Hypoglycemia does cause symptoms until glucose values are significantly elevated, usually above 180 to 200. So hypoglycemia means nope, 180, greater than 180. See, it's too low. And sorry, hyper. Next thing, am I the only one watching? Okay, glycolysis. Here we are touching a big fish. Okay, so we just drink little bit of water. Let's take a 5-minute break.